This is Chapter 6, A Tour of the Cell. So in this chapter, we will look at how the internal organization and structure of the cells allow them to perform and carry out these vital functions of life, right? Or those emergent properties that we talked about. So how do the individual parts of a cell come together as a whole to keep life functioning? Right? So we'll look at things like the membrane systems, internal membranes that operate with our energy and transformation of matter. So we can modify proteins, synthesize molecules. Chloroplasts can convert light energy to chemical energy. Mitochondria break down glucose molecules to form ATP energy, chemical energy. We'll look at how the cell can interact with the environment through the plasma membrane and the cell wall, and some extracellular structures. How the internal membranes are going to divide the cell into different compartments where specific functions are carried out in specific regions or structures in the cell. We'll look at genetic information storage and transmission through the nucleus. So how do we translate the information in the DNA into functioning protein? And then how do we make those proteins, right, with the ribosomes? So how do all of these individual parts come together to make the whole of a functioning cell? So because cells are usually too small to be seen with the naked eye, we have to use microscopes to help us study and learn about these cells. So standard type of a light microscope, which is what we have in our lab, is where visible light is going to be passed through the specimen and then through glass lenses, right, or different objectives. So these lenses work just to refract or bend the light to help magnify and blow up that image that we're looking at. So there are three important parameters to microscopy, right, that we need to consider. So magnification is basically the ratio of the object's image size to its real size. So how much is that object blown up or magnified? So basically, we just want to make sure that the image is large enough that we're able to see it with our eyes and make out the details. Resolution or resolving power is the clarity or sharpness of the image. So how are we able to focus in on the fine details of what we're looking at? Even if we magnify something, if it's blurry or out of focus, it's not going to do a whole lot of good if we can't make out what it is that we are seeing. Finally, contrast. So we need to see the visible differences in brightness in different areas of our sample. So we need to be able to distinguish different parts of our image from the background, right, and have that contrast between the two. So your typical light microscope, like what we have in our labs, can magnify up to about 1,000 times the size of an actual specimen. So usually we use the highest power objectives, the 1,000 times magnification for the smallest cells like bacteria. So there's different techniques that you'll learn in lab to help you enhance the contrast, to be able to see and focus on different parts. You'll also see that some specimens can be stained in different colors to help enhance that contrast. So a lot of, say, the tissue or cell slides that we look at in lab have been stained just so they stand out against the background and we're able to see their components more clearly. The resolution of our standard light microscopes is generally going to be too low to study the organelles or the small components in the cell or eukaryotic cells. So here is showing light microscopes. Right, we can see some bacteria on the highest power, but really the only organelle or component of a eukaryotic cell that you'll be able to see with our standard microscope would be the large nucleus in the middle of the cell. So you won't really be able to see things like the mitochondria or some of the smaller organelles or structures inside the cell. So if we want to look at even smaller or more detailed structures, there are two more high-powered type of microscopes that we can use, which unfortunately we do not have on our campus. But the two basic types of electron microscopes are used to study these subcellular structures, so things that are much smaller than an individual cell. Scanning electron microscopes, or SEMs, are going to focus a beam of electrons onto the surface of a specimen. So we're looking just on the surface, the outside of our specimen, but it gives us these really cool kind of 3D images. Whereas a transmission electron microscope, or TEM, is going to focus the beam through a specimen. So we're kind of looking at like a cross section. So it's going to transmit through instead of just scan the surface. So this would be if we're looking to study the internal structure of a cell. So in this example, here's our standard light microscope view of pollen, like our microscopes in our labs. This is the scanning electron microscope view of the pollen and a transmission electron microscope view, where we're doing kind of like a cross section of this pollen grain. Some more recent advances in the field of microscopy include labeling individual cells or components with these fluorescent markers, just to improve the level of detail that can be seen. So we can actually see some of the individual components and different fibers and things that make up the cell. So here we're looking at some mitochondria, some actin, some protein filaments, and some DNA in the nucleus. Another type of advanced microscopy is confocal and deconvolution. So just to provide much more sharper three-dimensional type of images of these cells and tissues. Cytology is the study of cells. Whenever we see cyto, we're talking about cells. So remember back in chapter 1, we talked about cell theory, which basically says that the cell is the basic structural and functional unit of life. So every organism is composed of cells. All cells come from pre-existing cells, and all organisms are made up of one or more cells, which could be prokaryotic or eukaryotic. So either they have a nucleus or they don't. So when we talked about the 3 domains of life on the evolutionary tree, the only organisms that are prokaryotes are the bacteria and the archaea. Right? So these are single-celled prokaryotes, very kind of simple, more primitive type cells. So the eukaryotes contain a nucleus, so our DNA is enclosed and protected within the nucleus. So we have more advanced types of organisms and cells and have multicellular organisms. So this includes protists, fungi, plants, and animals. So even though prokaryotes and eukaryotes are different types of cells, different branches on the evolutionary tree, all cells, both prokaryotic and eukaryotic, do share a few of the same basic common features. So what we do all have in common is all cells contain a plasma membrane, right? So we have to have kind of that boundary to separate the internal cellular components from the outside environment. All cells contain cytosol, which is the fluid portion inside the cell where all of the cellular components will be kind of suspended. All cells have chromosomes because all cells have to have genetic material, right, to reproduce. And all cells contain ribosomes. So we'll talk about ribosomes in this chapter, but they're basically the organelle or the part of a cell that makes proteins. So all cells require proteins, so all cells will contain ribosomes. There is a slight difference in the type of ribosome between prokaryotes and eukaryotes. Basically, eukaryotic ribosomes are a little bit bigger, but they serve the same function in making proteins. So what characterizes prokaryotic cells versus eukaryotic cells is, we said the main feature was the lack of nucleus. So prokaryotes, bacteria, more simple, primitive cells, they don't have a nucleus. Their DNA is just kind of free floating in the cell in what's called a nucleoid region. They have no membrane-bound organelles, right? So no complex little organs or structures inside their cells. And they typically will have their cytoplasm bound by a plasma membrane and a cell wall. So not all eukaryotic cells will have a cell wall. The eukaryotic cells are characterized by having a membrane-bound nucleus that contains the DNA. They'll also have some membrane-bound organelles. So the cytoplasm in a eukaryotic cell is bound between the plasma membrane and may or may not have a cell wall. So animal cells, human cells, we don't have a cell wall, but some plant and fungal cells do. And generally, eukaryotic cells are much, much larger than prokaryotic cells. So this is a picture of an epithelial cell. So say the inside of your cheek cell, right? You can see the nucleus, this large central organelle. These little dots or specks on this epithelial cell are bacteria. So even though we have different types and shapes and sizes of cell, there are some upper limits on how big a cell can be. So why don't we have, basketball-sized cells? Well, because the metabolic requirements and the principles of diffusion are going to limit our cell size to kind of the ideal ratio. We said the plasma membrane is that selective barrier that's going to separate inside cellular components from the outside environment. So it's selective in that it allows only certain substances in and out of the cell. So things like oxygen, nutrient, and waste. So the surface area to volume ratio of a cell is going to be critical in making sure that it receives adequate nutrients via diffusion. So as the size of our cells increase, its volume is going to grow proportionally more than the surface area. So in this example, we have a 1 centimeter cube, a 2 centimeter cubed, and a 3 centimeter cube. So the dye, the pink dye, diffuses inside our gelatinous cell model that we've made here. The pink material, the dye, is only going to be able to travel about 1 centimeter inside of this membrane, right, this gel. So if the cell is small enough, it can completely saturate and reach entire internal contents of our cell. If the cell is too big, so that more internal components of the cell aren't going to be able to get their nutrients and things via that diffusion process. So the process of diffusion is faster and more efficient over shorter distances. So as distances become larger, it has to travel farther, right? Then it's going to slow down that process, become more inefficient. So this is showing one cell, right? So one cube-shaped cell. So say we have a total surface area of 6, right? So total volume of 1. So our surface area to volume ratio is 6. Right? If we were to increase the size of this cube by 5, right, so now the total surface area is 150, right, total volume is 125, and our ratio has gone down to 1.2. However, if we break this down into 1 centimeter cubes, right, we keep the same volume, right, but increase our surface area. So we increase that ratio back to the original level. So now all of the individual cells are able to obtain what they need from the diffusion. So eukaryotic cells are a bit more complex in that they have internal membrane systems that are going to divide the cell into different compartments called organelles. So the organelles are similar to our own organs. So organelles, think they're little organs that each play their own specific role and function in keeping the cell alive and functioning. Plant and animal cells are the two most common type of eukaryotic cells, and they have most of the same organelles with just a few differences. So they all have ribosomes, right, because they both have to make proteins. They'll both have endoplasmic reticulum, plasma membrane, Golgi apparatus, mitochondria, nucleus. Where plants differ is that they do have a more rigid cell wall, and they contain a central vacuole, right, to contain water, and they have chloroplasts for photosynthesis. Whereas animal cells have small storage vacuoles or no vacuoles. They have what are called centrioles and centrosomes to assist with cell division, and we'll look at later. And some may have flagella, or those little whip-like tails, to help propel them through fluid solutions. So we said the nucleus was kind of the hallmark characteristic defining characteristic of a eukaryotic cell. The nucleus is going to contain most of the DNA within the eukaryotic cell. We said before, the DNA is basically all of the instructions and recipes for all of the proteins that make up a cell in an organism. So the ribosomes are the organelles, or the part of the cell that's going to translate the information that's in the genetic code in the DNA to make those recipes and make those proteins. So the nucleus is the largest organelle, the most conspicuous. So it's generally the only organelle or cellular structure that you'll really be able to see under our standard light microscope. So it's typically kind of the dark dot in the center of a cell. The nuclear envelope is going to enclose the nucleus and separate it from the cytoplasm. So the nuclear envelope consists of a double membrane, right, that's composed of a lipid bilayer, similar to the plasma membrane. So this nuclear membrane is lined with pores, a pore complex, just to help regulate entry and exit of molecules from the nucleus. So very similar again to the cell membrane, it's going to be selectively permeable. So we don't want just anything having access to our DNA. So when we transcribe a gene into messenger RNA, it can then exit the nucleus through the nuclear pores. The nuclear lamina is just going to be composed of these proteins that help maintain the overall shape and structure of the nucleus and the nuclear membrane. So within the nucleus, the DNA is organized into discrete units called chromosomes. So we're used to seeing DNA in these nice little compact X-shaped chromosomes. But really, each chromosome is going to contain one DNA molecule that is going to be coiled and wrapped around proteins. So altogether, this is called chromatin. So normally when the cell is not dividing, it's just carrying out its normal cell business, the DNA is just kind of loosely unraveled, kind of like spaghetti noodles, just all in the nucleus of the cell. So that way it's easy to access those genetic codes, those genes, to make RNA and synthesize proteins. It's really not until the cell gets ready to undergo division. So when we want to make a copy of a cell, make a copy of the DNA, when we package it down into these tight X-shaped chromosomes. So that way we don't leave any DNA behind during the division process. So the chromatin condenses down by wrapping around histone proteins and coiling and then forming supercoils until it gets into these tight X-shaped chromosomes, like I said, as it prepares to divide so that we don't leave any DNA or any genes behind. Within the nucleus, you have another central core called the nucleolus, which is going to be the site of ribosome synthesis. So we said ribosomes are going to make the proteins that are coded for in the DNA. So the nucleus contains the instructions for the proteins, and it also contains basically the instructions for how to make the ribosomes that make the proteins. Ribosomes are the protein factories of the cell. So they are complexes composed of ribosomal RNA and protein. So remember before we talked briefly about the three different types of RNA. So messenger RNA is what's going to carry the message encoded in DNA. Ribosomal or rRNA is going to compose the physical structure of the ribosomes. And tRNA or transfer RNA is going to deliver the amino acids to the growing polypeptide chain. So there are two types of ribosomes, or two locations where they can build their protein. So they can be free ribosomes, where they're just kind of floating in the cytoplasm throughout the cell, or they can be bound ribosomes, where they're bound and stuck to the rough endoplasmic reticulum. So we mainly use the free ribosomes to make proteins that we're going to use within the cell. So we'll make the proteins from the ribosomes, and then they'll stay kind of in the cell to be used locally. Bound ribosomes on the rough endoplasmic reticulum are going to make proteins that enter into this assembly line for secretion or transport out of the cell for use elsewhere. So the bound ribosomes are part of this endomembrane system. It's going to consist of the nuclear envelope, where the genetic instructions are found. We're going to pass through, enter into the endoplasmic reticulum where we have these bound ribosomes. So the proteins made by these bound ribosomes will then be packaged by the Golgi apparatus into vesicles or lysosomes, where then they can bud out of the plasma membrane and travel wherever they need to go. So this endomembrane system you can think of as regulating the protein traffic that helps perform different metabolic functions. So these components can be continuous, or they can be connected or transfer their components through these vesicles, right? Because they're all composed of kind of a lipid membrane, right? So they can bud off into small membrane sacs to package and carry different products. So once the mRNA has been copied from the DNA, it leaves the nuclear pore, right, crosses the nuclear envelope through a nuclear pore and enters into the endoplasmic reticulum. The endoplasmic reticulum accounts for more than half of the total membrane area in many eukaryotic cells. It's going to be continuous with that nuclear envelope, again, because those ribosomes are going to start translating that mRNA as soon as it leaves through the nuclear pores. There are two distinct regions of the endoplasmic reticulum. So the smooth ER is called smooth because it doesn't have ribosomes, right? So it doesn't have that rough, studded appearance because it doesn't have those ribosomes. So the smooth ER's function is to synthesize lipids. It also detoxifies drugs and poisons and can help store calcium ions, like in muscle cells. One type of cell that you may expect to find large quantities of smooth ER would be, say, in the liver, right? So the liver is our body's detoxifying organ. So liver cells have lots of smooth ER to carry out that function. Whereas the rough ER is rough because it's studded or textured with all of those ribosomes. So all those ribosomes are going to function to make our proteins. Once the proteins are synthesized by the ribosomes in the rough ER, they can then be sent to the Golgi apparatus, which is kind of like the post office of the cell, right? So it's our cell shipping and receiving center. The Golgi apparatus consists of flattened membranous sacs that are going to modify, sort, and package these newly synthesized proteins and materials into our transport vesicles, right, where then they can be secreted out of the cell. Lysosomes are one product that we can synthesize coming off of the Golgi apparatus. They're basically like the digestive system of the cell, so little stomachs, little bags of digestive enzymes. Proteins synthesized by the ribosomes on the rough ER are going to be digestive enzymes. Right? Remember, enzymes are just proteins that help facilitate chemical reactions. So now these enzymes are going to be packaged into a vesicle, or little membrane sac, transported to the Golgi, where the Golgi will then modify and pack these enzymes into a lysosome. So this cell has taken in some food particles by endocytosis. So now we have a food vacuole, so a little sac for food. So this would be the stomach. We have our digestive enzyme vacuole that can fuse with the food vacuole. So now these digestive enzymes can start to break down these food particles. This structure would be referred to as a phagolysosome, where we have the fusion of a lysosome and a food vacuole. So the process of phagocytosis is just cell eating. It's also sometimes referred to as endocytosis. So we're bringing something inside the cell. We're eating something, bringing it inside. Lysosomes can also be used to break down and recycle old or defective cell parts and organelles, just to keep the cellular internal contents clean and tidy as things break down. So this process is called autophagy. Vacuoles are diverse maintenance compartments or storage containers of the cell. So they can be derived from that endoplasmic reticulum and the Golgi apparatus. So essentially little membrane sacs that can store different substances for the cell. So freshwater protists can have what's called a contractile vacuole, where they can store water. Plants have large central vacuoles that have to do with the turgor pressure of the cell, or keeping the plant from essentially wilting. So here is a plant that has full central vacuoles, Right? So when the central vacuole is depleted of water, doesn't have enough water, right, it shrinks the internal contents of the cell and causes the plant to wilt. Food vacuoles are formed by that process of phagocytosis, or cell eating, when it's bringing in food particles from outside the cell. Some vacuoles can also be a repository or a storage container for ions to be used later on in some biochemical reactions. Mitochondria are the sites of cellular respiration that's going to generate metabolic energy in the form of ATP. So using glucose sugar and oxygen, we can chemically convert that into adenosine triphosphate that then the cells can use to power their own processes. Chloroplasts are found in photosynthetic organisms like plants and algae that are going to carry out photosynthesis. So they convert light energy and carbon dioxide to make sugar and oxygen. So chloroplasts and mitochondria are very closely related because they're both part of what's called a coupled reaction. So the reactants and products of photosynthesis, right, so sugar and oxygen in photosynthesis, is then used as the reactants in cellular respiration by the mitochondria. Right, so then the products, the end products of cellular respiration, CO2 and water and energy, is basically going to be the ingredients or reactants for photosynthesis. Right? And we have kind of this nice cycle between the two sets of reactions, what's called a coupled reaction. Mitochondria and chloroplasts are also interesting of the organelles because they do have some similarities with bacteria. Both are going to be enveloped by double membrane. They have their own ribosomes. And the ribosomes are the smaller type that are more commonly found in prokaryotes. Remember we said eukaryotes just have larger ribosomes. Well, mitochondria and chloroplasts have those small type of ribosomes. They also contain their own DNA in a circular chromosome, which again is only found in prokaryotes. So if you've ever heard of mitochondrial DNA, that's only passed from the mother, where they can trace back your maternal lineage through the mitochondrial DNA. So that's because for fertilization, the female egg is going to contain all of the organelles and the cell components, and this is a larger cell. Whereas the male sperm basically is a tiny cell that only contains the father's DNA. There are no other organelles really that contribute to the development of the zygote. So the only mitochondria that the embryo obtains is from the mother's egg. Another interesting similarity with mitochondria and chloroplasts to bacteria is that they can grow and reproduce independently in the cells. So cells are able to kind of make more mitochondria or chloroplasts as needed. So these similarities led to what's called the endosymbiotic theory of eukaryotic evolution, which basically suggests that a very early ancestor of eukaryotes engulfed a smaller oxygen metabolizing prokaryotic cell. And over a long period of time, they became dependent on one another and formed a symbiotic relationship. They could no longer live independently, right, becoming an endosymbiont. We have a large prokaryotic cell that ingests a smaller aerobic oxygen-using bacteria. The small bacteria gains a safe place to live, right? Doesn't have to worry about getting eaten, has some nutrients provided for it. The larger prokaryote is going to obtain ATP energy from the smaller one. Over a long period of time, they became the mitochondria, and they couldn't live on their own anymore. So they just became a part of the larger cell. And then this process happened again, but with a photosynthetic cyanobacteria. So now the cyanobacteria endosymbiont is going to produce sugar for the larger cell. So this is where we had the evolution of plant cells. So there's been some recent studies showing that maybe the idea that this has happened again with some other organelles like the nucleus. So it was actually just last semester in my general biology, we were discussing how there was a paper discussing how viruses, when a virus infects a cell, it makes kind of a pseudonucleus to contain all of the genetic material of the virus and assembly for parts of the virus. So there's a theory out there now, they're looking at maybe the nucleus began as an endosymbiote through a virus infection. Something interesting to think about. So nearly all eukaryotic cells will contain mitochondria because all cells require ATP energy. So we said mitochondria have a double membrane structure. So they have a smooth outer membrane and a folded inner membrane called cristae. So these folds are called cristae. So these are basically just to increase the surface area. So we have more surface area for cellular respiration chemical reactions to occur. So the more surface area we can obtain to carry out the chemical reactions, the more ATP we can generate in that space. Chloroplasts contain the green pigment chlorophyll, as well as some other enzymes and molecules that function in photosynthesis. Right, so what gives plants that green color is the green pigment in the chloroplast, the chlorophyll. So later on we talk about photosynthesis, we'll talk about why it's green, but it has to do with wavelength energy absorption. So the green is going to be the most ideal for absorbing the correct amount of wavelength light energy for the plant cells. But chloroplasts are found in the leaves and other green organs and structures of plants and in algae. So the parts of the chloroplast include the thylakoids, which are basically these little stacks of green pancakes, little membranous flattened sacs. And then the stroma is the fluid portion inside the chloroplast. So we have the chlorophyll packaged in the thylakoid. So this is where the photosynthesis reaction is going to occur. So it's folded and stacked like this, similar to the cristae in the mitochondria, just to increase our surface area for these reactions to occur. And then you can have many chloroplasts kind of bonded together in a larger organelle called a plastid. We talked about lysosomes earlier being kind of like the digestive stomachs, digestive organs of the cell. Well, peroxisomes are just a specialized type of metabolic organelle that's similar in structure to lysosomes. So we still have kind of this vesicle, this membrane sac of enzymes. So whereas lysosomes are filled with kind of digestive enzymes to break down substances, peroxisomes are filled with enzymes that help to detoxify hydrogen peroxide and other harmful compounds like alcohol in the liver. Right, so we said the smooth ER, the smooth endoplasmic reticulum, helps to detoxify substances. So liver cells would have lots of smooth ER. They would also have lots of peroxisomes to detoxify alcohol. The enzymes in peroxisomes also help play a role in the oxidative breakdown of fatty acids in animal cells. So this is how we break down stored fat to use for energy in cellular respiration. So when you start to burn fat, right, you're breaking down that fat, those fatty acids to use for ATP synthesis. The cytoskeleton is similar to our own skeleton. So it gives the cell its overall structure and support through a network of different protein fibers that extend throughout the cytoplasm. The cytoskeleton helps to organize the cellular structure and activities while anchoring some of the organelles. So we don't have them just float away. We can kind of keep them in their respective regions in place. So there are three types of molecular structures that comprise the cytoskeleton. So things like microtubules, which are little tubes or proteins called tubulin, microfilaments like actin, and intermediate filaments, which is kind of in between these two. So microtubules are essentially hollow tubes composed of tubulin protein. They help to maintain cell shape, cell motility. They're also going to contribute to chromosome movement during cell division. So we'll talk about a little bit later the spindle fibers, right? So these microtubules are going to attach to the chromosomes and position them in the right location during cell division. So that way we don't have an unequal division, right? So that's divided straight down the middle of the cell. Microfilaments help to maintain the cell shape. They also contribute to some cell motility or movement. So when we have muscle contraction, we'll talk about later, our muscles contract via action of these different microfilaments. Cytoplasmic streaming, or ability to move fluid and substances around inside of a plant cell, are also due to the actions or movements of these microfilaments like actin. Intermediate filaments are more just to kind of maintain the cell shape. So they don't have as much kind of secondary functions as microtubules or microfilaments. So this is showing part of the cytoskeleton, and we have a motor protein that is transporting a vesicle, right, that's going to contain maybe a new protein or something to deliver it where it needs to go. So the cytoskeleton helps to support the cell maintain its shape and interacts with these motor proteins to produce cell motility. So it provides kind of like a highway for some of these motor or movement proteins to operate on. So inside of a cell, vesicles and other organelles can use these motor protein feet to travel along these tracks provided by the cytoskeleton. So this is a computer animation of what it really looks like in the cell. So we have these protein fibers and filaments and tubes, and then these motor proteins that literally kind of walk along attached to these tubules while carrying these vesicles throughout the cell. So while it may look like you're not doing much on the surface, at the cellular level, we're all very busy. So in animal cells, microtubules are also going to function in cell division. So they're going to grow out from the centrosome and form these spindle fibers from the centrioles in order to, like I said, attach to the chromosomes and position them in the correct location during cell division so that we have a equal division of the nucleus and the chromosomes. So these centrioles that form these spindle fibers are generally arranged, these microtubules, in 9 triplets. So they come in sets of 3. So you have 9 sets of 3 kind of forming almost this kind of star-shaped structure. And this is showing the spindle fibers emerging from those centrioles, right, these microtubules, and then grasping the chromosomes and positioning them along the midline equator of the cell. So we have an equal separation of chromosomes. Microtubules are also going to help control the movement of flagella and cilia. So these are cellular extensions that help move cells through a fluid or move fluid around a cell. So a lot of unicellular protists and even human sperm cells, right, are going to be propelled through fluids by these extensions. So generally, cilia are shorter little hairs on the surface of a cell or an organism that help to circulate fluid around the cell, whereas flagella are more single, long, whip-like tail extensions that help propel the cell through a fluid, help it swim through the fluid. So this is showing an paramecium, right, that is beating fluid around it, trying to get some nutrients. Our respiratory tract is actually lined with ciliated cells as well, right? So it helps to keep. So the mucus in our respiratory tract is going to work to trap dust and debris, particles and things from getting down into the deeper respiratory system. And then these cilia that line your respiratory tract will sweep that mucus up and out of the respiratory tract. So even though cilia and flagella are functionally different, they do share a common kind of base structure. So we have kind of this bundle of the microtubules sheathed in some plasma membrane, some lipid membrane, and we'll have 9 pairs of microtubules arranged in a ring with a single pair in the center. So it kind of rotates and cranks a lot like a motor. So the basal body is going to anchor it to the cell membrane, And then there's a motor protein, so one of those little walking proteins that's going to drive the bending movements, right, and start the rotation of the cilia and the flagella structure. Microfilaments, so things like actin and myosin in muscle cells, are going to function more in our cellular motility. So our muscle cells contain actin and myosin that contribute to muscle contraction, but it also allows other cells to have different types of movements. So like an amoeba or single-celled organism, they're able to crawl by extending their pseudopods, right, or their false foot. So pseudo means false, podia, pod, I think podiatrist means foot. So they can extend certain regions or these pseudopods out and kind of drag the rest of their cellular bodies along. Right? They can also use these pseudopods to reach out and engulf food or prey items. Cytoplasmic streaming occurs in plant cells, so it's how plants can use the motility of these microfilaments to circulate the fluid or cytoplasm inside their cells. Intermediate filaments are kind of intermediate in size, so they're larger than the microfilaments and smaller than the microtubules. But whereas microfilaments and microtubules had those secondary functions for motility or maybe help regulate cell division and flagella movement, intermediate filaments are basically just permanent cytoskeleton fixtures. Right? So they help support overall cell shape and fix the organelles in place. So this is one of those fluorescent microscopy pictures of showing keratin filament. So keratin is the protein in our skin, hair, and nails that makes it very durable. Right? So these are all the protein keratin filaments forming the overall structural network of an epithelial or a skin cell. So we talked about structures inside the cell, but there are some extracellular components and cellular connections between cells that can also help coordinate our cellular activities. So most cells will synthesize and secrete material outside of the cell as well. So these structures can be involved in many essential cellular functions. So one example would be a cell wall. So we said the cell wall was one of the distinguishing features between a plant cell and an animal cell. But prokaryotes, like bacteria, also have a cell wall. Fungi contain a cell wall made of chitin, right? And some protists can also have cell walls. So even though the cell wall between a plant cell, a bacteria cell, and a fungal cell are composed of different types of molecules, they're going to serve the same type of function and just an added layer of protection and support and rigidity and structure to the overall cell. So the cell wall in plants is going to help protect the cell inside, maintain the shape, and prevent excessive water uptake. Plant cell walls, we'll just focus on for this chapter, are composed of cellulose fibers. Remember we said cellulose was one of those polysaccharide molecules. So we have these long chain polysaccharides that are arranged kind of like a raft structure and sandwiched between some other polysaccharides and proteins, right, and the cell membrane. So here's our bilayer, phospholipid bilayer, and our cellulose, and kind of sandwiched between this middle lamella layer. So plant cell walls may have multiple layers. So the primary cell wall is going to be the outermost region of the cell wall. It's relatively thin and flexible, so just that thin outer layer. As we go deeper into the cell, the secondary cell wall is going to be what's between the plasma membrane and the primary cell wall. Right? So it's this thicker portion, right, of the wall. The middle lamella is going to be the layer between the two walls, so kind of the space-filling area between adjacent plant cells would be the middle lamella. So primary, the thin outer layer, secondary, the thick inner layer, and middle lamella kind of in between the two and the space filling. So we said animal cells do not have a cell wall, but they do have an elaborate extracellular matrix. So extracellular just means outside the cell. So instead of having a cell wall, we can have different protein fibers, like collagen, which would provide some structural support to the outer surface of the cell. Proteoglycans, so proteins, and glycan meaning sugar, so protein sugars that can serve for kind of recognition and signaling. Surface molecules, so we talked about that a little bit before. Carbohydrate chains on the surface of a cell can serve kind of as identifying markers to other cells. Fibronectin, right, so this is a type of protein that can be part of the extracellular matrix that helps with adhesion or attachment to other cells and tissues. So these can bind to receptor proteins called integrins or integral proteins, meaning they're kind of integrated in the membrane. So components of this extracellular matrix can help regulate a cell's behavior by communicating with other cells and receiving and sending signals to other cells. So it can also help to influence the activity of gene expression within the nucleus by responding to different signals from other cells. So most cells in an organism are not going to be single, solitary, isolated cells. They're going to interact with neighboring cells and form tissues. So think back to those layers of structural organization. You have multiple cells working together, they form tissues. So neighboring cells and tissues, organs, and organ systems often are going to have to adhere or stick together, interact, and communicate with one another through some type of direct physical contact. So cell junctions in plant cells are called plasmodesmata. So these are just small channels that are going to connect the cell walls between different plant cells. So things like water, solutes, other chemicals and nutrients and things can pass through and be shared throughout the entire plant tissue. There are three types of cell junctions that we can see in epithelial tissues of animal cells. So think like your skin tissue. So epithelial cells are just covering and lining tissues. So three main types are tight junctions. So these are basically what they sound like. They're very tight connections. So kind of the cell membrane of adjacent cells is going to be stitched together very tightly to prevent any leakage of fluid. So like the tissue in the bladder epithelia has lots of tight junctions because we don't want any urine fluid to leak out of that organ. Desmosomes are more kind of like a Velcro connection. So the cells are attached, but they have a little bit more give and movement to them in the tissue than with the tight junctions. And then finally, the gap junctions are kind of like little pores connecting or little channels connecting adjacent cells that are more of a chemical connection than a physical connection. So these gap junctions allow neighboring cells to pass or share things like ions and chemicals for rapid intracellular communication. So one example would be in cardiac muscle in the heart, contains lots of gap junctions because we want all of the individual cardiac muscle cells to respond to the same signal to contract. So that way we have that coordinated kind of rhythmic heartbeat. We don't have a twitchy muscle contraction in the heart muscle. So as we've seen, none of the cell's components work alone. So we have kind of all of these individual parts coming together to form the whole of the cell. This example, a macrophage or a large eating cell. So its ability to destroy a bacteria cell is going to involve input and contributions from the whole cell and coordinating different components like the cytoskeleton. Right? So the actin microfilaments are going to have to extend those pseudopods to be able to engulf the bacteria, okay, so it can undergo phagocytosis and eat it. The proteins on the cell membrane are going to have receptors to recognize and bind to that bacteria to take it in, right? And then form the vacuole that contains the ingested bacteria. We'll have to synthesize digestive enzymes and package them into a lysosome. right, and then form that phagolysosome. So now we can digest that bacteria. And then we'll have to undergo exocytosis to dispose of the cellular debris or the leftover components after the digestion of that bacteria. So you can think of the overall cell kind of like a factory, right? So it takes in nutrients, right, and kind of the ingredients and components that it needs for its chemical reactions. The nucleus, right, contains the DNA, all the instructions. It kind of coordinates and directs all the other components. Our ribosomes are manufacturing our proteins, right, our endoplasmic reticulum making some lipids, some amino acids, right, fuels. The Golgi apparatus is packaging and sorting those products that we're making, right, for export, right? And then the mitochondria are able to generate energy to power the rest of the factory.